Hello students, so today in this video we will discuss about the seed. Okay, earlier we discussed about the fruit and now we will discuss about the seed. And of course, what we call the plants that produce seeds? Spermatophytes. Plants that produce seeds are called spermatophytes. So, what are the groups of plants that produce seeds in the plant kingdom? Okay. So, two groups are there, that is gymnosperms and angiosperms. And what is the difference? So, gymnosperms will produce only seeds, means their seeds are not covered by the fruit. That's why the seeds are naked seeds without fruit. That's why the technical term itself indicates they are naked seeded plants. Gymnos means naked, spermos means seed. And what about the angiosperms, which are called as flowering plants? Angios means vessel and spermos means seeds. So, what is that vessel like structure? The vessel like structure is the fruit. So, that indicates the seeds in angiosperms are enclosed by the fruit. That is why they are called angiosperms. In that way, we can differentiate gymnosperms and angiosperms, but both are having one common feature. What is that common feature? Both are producing seeds. That is why we call both of them as spermatophytes. Right. Then, what is the seed actually? How the seed is formed? And what structure in the flower will develop into the seed? Okay, already we discussed about the fruit, and there we defined the fruit as what? Post fertilized ovary. And what are present inside the ovary? Ovules. So, while the ovary is becoming the fruit, the ovules will become seeds after fertilization. So, we can define the seed as what? A post fertilized ovule is called seed. Post fertilized ovule is called seed. Okay. That means after fertilization, the ovule is changing into the seed. And when we examine a typical seed or a model seed, okay, what are the parts you will find in that seed? So, outside you will find a covering, and that is called seed coat, and a nutritive tissue will be there, and which is called endosperm, and the new generation is in the form of embryo, and in some seeds you will find some remnant nucellus which is called perisperm. And of course, I gave the ploid is here, and the seed coat is diploid, embryo is diploid, perisperm is diploid, but endosperm is diploid because it is the product formed as a result of triple fusion. Okay. So, now, and remember that how many seed coats are present in a seed? That all depends on the type of ovule. So, the ovules are of two types based on the number of integuments. Some ovules will have a single integument, and we call them as what? Unitegmic ovules. And those which are having two integuments are called bitegmic ovules. So, the integument is changing into the seed coat, after fertilization. That's why suppose if it is a unitegmic ovule became seed, then you will find only one seed coat, and that can be called as testa. And suppose if the seed developed from two integuments, then naturally the seed will have two layers or two seed coats, and one will be outer and the other one will be inner. And the outer one is called testa here, and the inner one is called tegmen. Like that. And of course, we know that in angiosperms, one characteristic feature is there, that is not a simply fertilization, double fertilization. And what is double fertilization? So, syngamy plus triple fusion. Syngamy results in the formation of zygote, and triple fusion results in the formation of pen, that is primary endosperm nucleus, and that primary endosperm nucleus develops into endosperm. So, what is this endosperm? A nutritive tissue or nourishing tissue, and it is formed to nourish the embryo. Okay, and now let us say the endosperm is also called as albumin. The other name of endosperm is what? Albumin. And now I wrote here two conditions you will find. By the time seed develops, by the time the seed is completely developed, so then this endosperm may be retained by the embryo. Okay, that's why in the developed seed you will find endosperm, and then it is called endospermic seed or albuminous seed, and these albuminous seeds are common among monocots. But of course, you remember one exception, there is one family in monocots, that is Orchidaceae family, in which endosperm is not at all formed. That's why Orchidaceae members will have non-endospermic seeds. The monocot family with non-endospermic seeds is Orchidaceae family. Orchids are the members of Orchidaceae. So now let us see the other condition. By the time the seed is developed, the endosperm is completely consumed by the growing embryo. So in that seed, you will find only the embryo, but not the endosperm, because that has been consumed by the embryo. So that is called non-endospermic seed. I hope you are understanding the point here. Generally, people think that the endospermic seed will have endosperm, and it is formed due to triple fusion, and non-endospermic seed is without endosperm, so there is no triple fusion. No, triple fusion is there. Endosperm is formed, but it is consumed by the embryo. So by the time the seed is formed, you won't find the endosperm. That's why it is called non-endospermic or exalbuminous seed. And such non-endospermic seeds are generally seen among dicots. But some exceptions are there. Exception among dicots is what? Castor. The castor seeds are endospermic. Okay, now let us say the embryo. That means the embryo represents new generation, new sporophytic generation. So the offspring is in the form of embryo, and that embryo will have one axis. The embryo will have one axis, and this axis is called embryonal axis, or the other technical term is what? Tigellum. And this embryonal axis will have upper portion which is called plumule, and the lower portion is called radicle, and at one particular point one or two cotyledons are attached. So these are cotyledons which are considered as first leaves of the plants. So this is what embryo actually. So the same thing I wrote here. So the axis is called embryonal axis or tigellum, and it will have plumule and the radicle, and we know that the radicle will develop into root system at the time of germination, while plumule will develop into the shoot system. And when it comes to the number of cotyledons, some will have one cotyledon, and we know that these are called monocotyledonous embryo. And monocotyledonous embryos are present in monocots. And when two cotyledons are present, the embryo is called dicotyledonous embryo. And these are present in dicots. So I hope now you are understanding the point that the two classes of angiosperms, dicotyledonae and monocotyledonae, are named after the number of cotyledons in the embryo. And sometimes you will find this nucellus. Actually, nucellus also gets consumed at the time of seed development, but in some cases you will find this perisperm, and the seeds with perisperm are called perispermic seeds. Perispermic seeds, and the example is black pepper and also coffee. Okay, now let us see the details of the structure of the seed, and here I drew the diagram of dicot seed. Okay, so this is chickpea or Bengal gram, and you can see here this is the external view of the seed, and you will find one small pore will be there, and which is the micropyle of the seed, and you will find one scar here, and that scar is the place where the seed is attached to the placenta, and that is called hilum. The place where the seed is attached to the placenta is called hilum, and it is represented by one mark or scar. That is what hilum. And what you will find outside, seed coat. And when we remove the seed coat, then inside you will find what the embryo, because it is dicot, so non-endospermic. So the two, okay, this is the embryonal axis. This is embryonal axis, and the upper portion is plumule and the lower portion is radicle, and these are two cotyledons. And I hope now you are understanding in this chickpea what we are eating, cotyledons. Means what in all pulses what we are eating are what the cotyledons. Okay, so that is dicot seed, and let us come to the monocot seed. And the example which I have taken here is maize or corn. Okay, and actually we are familiar with this corn, and generally we mistake the corn as what a seed. The corn is actually a fruit, and inside that fruit you will find the seed, and inside that seed you will find the embryo. Okay, so you know that the shape of this corn will be like this, and outside there is pericarp, that is fruit wall, and inside this is the seed. So the whole thing is what? Seed. And maize is a monocot. So in monocots you will find what? Endospermic. So that's why there is endosperm here, and this is the embryo, and the embryonal axis, scutellum. And what is the scutellum? That is the large cotyledon is scutellum. But for details of that, let us take a sectional view of the same seed. So this is the sectional view of the seed. And here you can see the pericarp once again, and this is the endosperm actually, but I am showing with some lines here. This is protein layer, and this protein layer is called aleurone layer. And remember that aleurone layer is a endospermic layer, but only thing is that it stores proteins. So what is the ploidy of this aleurone layer? Diploid. And actually in the endosperm will store starch, so starchy endosperm, that is also diploid. And this is the embryo which is diploid, and these are different parts, and we will find this is the embryonal axis behind which there is large cotyledon, and that large cotyledon is called scutellum. And this is the embryonal axis, and the lower part is called radicle, and the upper part is called plumule. But in these monocots, monocotyledonous embryos will have one covering. The covering present over this plumule is called coleoptile, and the cover that is present over the radicle is called coleorhiza. Okay, so this is the sectional view of the monocot seed. That is about the information about the seed.